Thanks, Tricia. Okay. There we go. Okay. Hopefully you can see my slides. So yeah, I really wasn't sure what to talk about here. I don't work on sedimentary DNA. I don't really do an awful lot of work on DNA. I have helped people with it. And also I've kind of, if you know anything about where I'm now working, I'm now a statistician essentially in the Department of Animal Science. And a lot of my work has gone sort of ecology based. I also didn't know how controversial to be. And I probably erred on the side of not being that controversial with critical transitions. And I'm focusing more on the methods that we use. And maybe this is a sort of plea to people to maybe try new things, try statistical models for your data, rather than necessarily using the ad hoc or metric-based approaches that have perhaps predominated in some earlier work in these areas. And so I think the old bit was me just talking about, well, how have we traditionally done these things? And then the new bit is how might we do these things now? So just land acknowledgement. This work was really, I mean, it's just a collection of thoughts, to be honest, and a bit of our code and a bit of time sat in front of the computer. But it was started when I was at the University of Regina, and the university sits on Treaty 4 lands, which these are the territories of the Nihiewuk, the Cree, the Anishinaabe, the Saulteaux, the Dakota, Lakota, Nakota, and it's also the homeland of the Métis Nation. And then also the pigment data that I'm going to be talking about briefly from Lake 227 that were collected by Peter Levitt, my colleague at the University of Regina. That lake is in the experimental lakes area in north-west Ontario, and that is an area that is on Treaty 3 land, sorry, and that's the territory of the Anishinaabe. My work in Canada was funded by NSERC through a Discovery Grant and also by support for students from our Faculty of Graduate Studies and Research. And I now have an Aarhus University Research Foundation starting grant that is sort of supporting some of this work as well. And yeah, and I've already put the link to the slides in the chat, so you can go and have a look at those if you want. Okay, so one of the things I wanted to think about was something a bit more generic than early warning indicators, because I don't always think that we can easily look at ecological transitions of the critical transition type from paleo data for a lot of reasons that we can maybe get into over a beer at some other time, COVID allowing. But I want to talk more generically about indicators that we might estimate from paleo time series. So one of the ways in which the critical transitions have been presented as a model is through this idea of alternative stable states, that under a range of conditions, multiple ecosystem states may exist. We might describe these states through a stability landscape. These depict the, I'm just going to see if I can turn on the, here we go. Where's the spotlight? There we go. So hopefully you can sort of maybe follow my pointer as well. Okay, so they've been described by these sort of landscapes where we have a system that looks like these valleys and hills, and the ball is the ecosystem. The states, the different stable states are these individual basins or valleys, and the ecosystem sort of bounces around in these things, and if it gets a big enough kick, you might kick it from one basin of attraction to another. But we might envisage damage to the ecosystem as altering the shape of the stability landscape. So here we may be in a situation where only one stable state is present. But as we degrade the ecosystem, we end up with having multiple stable states available. And then eventually, as we degrade the system far enough, the ball can get kicked over into another state. And this is actually a useful toy diagram of thinking about how these things work. But we don't necessarily believe that this is the exact mechanism. It's just a way of describing these things. Now, the variance is often known as an early warning indicator of this state change, moving from one basin to another. And in this diagram here that I took from Madden Sheffer and co.'s paper in Science some years back, we can illustrate why variance is important. So in panel A here, we've got a high resilience system. This is a very steep-sided valley. If the ball gets knocked around in here, it's going to roll very quickly down to the bottom of the valley. And so we see that in the measure of the system state here. The dashed line is its equilibrium. We perturb it from its equilibrium. It returns very quickly to that equilibrium. There's some oscillation, but it essentially settled down around the equilibrium again very quickly. And if we sample data from this kind of system, this is kind of what we'd expect. There's very little autocorrelation in this system because it's essentially a series of random shocks. Converse is a system where we have low resilience, where we've eroded the shapes of these basins, the slopes of these basins, so that the ball rolls very slowly back down to the equilibrium point when it's been disturbed. This is illustrated here. We give the system a kick. It takes a long time for the system to transit back down to being close to the equilibrium, and then it kind of oscillates around for a while, okay? And if we look at data from that kind of system, if we simulate data, then it would look like this panel here, panel F, where we have what essentially look like stochastic trends. There's a lot of autocorrelation in here to generate those stochastic trends, but there's also more variance. And so this is where this idea of these indicators comes from, is that as the system approaches this low resilience state, maybe we start to see this increased autocorrelation, but also an increase in variance as we approach a transition. But maybe we can just use variance as a general indicator of resilience and not necessarily something that is associated with critical transitions. Now, in ecology, resilience has basically got too many definitions. It's an overused word and people have redefined it many times. I'm just going to define it as the time required for an ecosystem to return to an equilibrium or steady state following some perturbation. And given that definition, there's nothing implicit in there that the system must have fold bifurcations or be a hop bifurcation or some kind of model of a critical transition. It's just a measure of how quickly the system returns to some equilibrium. And some ecologists would define that as stability. Holland would refer to this as engineering resilience, right? But even if you don't buy any of that, maybe you can buy into the thing that the variance, the spread of our observations around some mean is useful, even if it's just useful phenomenologically. Try and say that fast many times. Maybe it is, and that's what I'm working from here, that this might be something that's useful even if we don't buy into the idea of that we can estimate alternative stable states and critical transitions in a data series, just from the data themselves. So how do ecologists compute the variance? Well, they use metrics, right? So a variance of a time series, obviously it's really easy to compute the variance of a data set, but how do they do it for a time series? Well, they use a metric, and that's a bad thing, I believe. Ecologists will get some number and that'll be great. They go, wow, we've got a number and then we're going to do something with it. And then they want to know whether that's an important number. Is that a high amount of variance? Is that a low amount of variance? Is there a trend in that variance? And so then they typically apply brute force to that. They get the computer to do lots and lots of calculations and we get some number out of it. And I've done that. I am an ecologist, so I do that. I'm also a statistician. And I want to know what are the properties of these metrics, these novel estimators that they've developed. And typically when you start looking into these estimators that ecologists develop and looking at what they're doing with that brute force inference, typically the whole process is then fraught with loads of problems. And it would have been better if we'd started from a more traditional statistical model than if we'd just started from the metric and tried to bolt some inference on top. So the example I want to talk about, and I've talked about this at a conference before, is this looking at early warning signals of a critical transition using a moving window to estimate variance. But the same problem applies to the autocorrelation as well in these kinds of data. So we've got a data set up here. I'll talk more about what this data is in a minute. We're going to the process of looking for or evaluating how the variance is changing in this system. The recipe is to detrend that in some way. So the red line here is a low dimensional smoother that's put through the data to take out the gross trend. That gives you these residuals because you subtract the fitted value of the trend from the data point, gives us residuals that essentially have no trend. Now we can look at the spread of these points around the zero line. Essentially, that's what we're doing. So they do that using a rolling window. So they take a window width, so you have to choose how big a window to use. Here, the window width was set to be half the observed time series. And then you take all the observations in the window, you calculate the variance and note it down. You move the window along one time step. You've got all the observations now that are in that new window, and you calculate the variance again. And you move the window along one step per time, and you calculate the variance. And that gives you one of these time series in the bottom here. And because of the way they did the rolling window, they had to observe half the record, the time series, before you could get this trend of variance. And then they say, well, okay, let's look at this new data set. Is there a trend in this new data set? And they think, well, let's use Kendall's rank correlation because this doesn't look linear and various other things. So they use Kendall's non-parametric trend test, and then they can say something about the significance of this increasing variance. Okay. Well, that's great. And this figure itself comes from Dacos-Sedell, but we have problems actually applying that to ecological data, anything other than ideal data. And we certainly have big problems when it comes to applying this kind of idea to paleo data. So the first thing is, how do you detrend this? Yeah. If you over detrend it, you're going to perhaps remove signal that should be something that you're looking for in the variance. If you don't remove enough signal, if you don't detrend the signal well enough, to just get something that has no trend, then you will ascribe to the variance something that should be ascribed to the trend, right? So you get biased estimation. But how do you do that, right? What window width? How wide should this window be? All right. What about this works for regularly spaced data because you're moving the window along one time step. Well, most paleo data are not regularly spaced. So what do we do about gaps or missing data in any kind of time series? But especially in paleo, where the data are just not regularly spaced, right? Well, you can try and do things with that. And some people did that and got into a bit of trouble doing that because the process that they used to sort of standardize their data could also induce the very patterns that they were trying to look for. Right? Every time you have to manipulate your data in some way, you could be inducing some artifacts into it that could bias your results. And then the final problem is that statistical testing is hard. Kendall's tau assumes independence, your observations are independent. And because of the way the moving windows work, these are essentially a moving average of the variance in a sense. And we know, therefore, that they have a complex dependent structure. So the theory behind Kendall's tau breaks down. You cannot compute a p-value for it. You might be able to use brute force inference using surrogate time series, but then they do that by fitting an ARMA, an autoregressive moving average model, to these residuals. But why is an ARMA the correct model to fit to these residuals? What about any other model? And then ARMA requires us to fit the model to data, and typically those require the data to be regularly spaced. So we have another problem. All right? So by going down this route of choosing the moving window, it's generated a cascading series of problems that need to be resolved. All right? And it would be better if we could maybe take a step back. Let's look at how we might approach this from a statistical point of view, and then avoid some of this ad hocery. And one way we could do that is through what's known as a distributional model. So typically we have focused on the mean when we fit our regression models. We fit a linear regression, that is a regression model for the mean. But we typically forget about the variance. In fact, in a linear regression, The variance is just something that is an afterthought. We just assume that it's constant and we estimate it from the residuals after we've estimated our linear regression line, for example. Okay? But we don't have to do that. We could actually fit models for the mean and the variance. And we could do this for all kinds of distributions. And it's really easy for the Gaussian, or it's easier for the Gaussian, because the Gaussian distribution has two parameters, a mean and a variance, and they are independent of one another. So we can change the mean, as in this left panel here. These two distributions have the same variance, but they've got different means. Likewise, here on the right, I've got two distributions that have the same mean, but they have different variance. So rather than assuming that the variance is fixed in our regression models, we can maybe model that variance as well as modeling the mean. And then we don't have to worry about detrending first, then looking at residuals. So now we've got a cascade of models that we have to compare. We're going to fit one model that has both the mean and the variance in the same thing. So how do we do that? Well, we're going to say that our observations, given our covariates here, have some distribution. That distribution is parameterized by two or more parameters, or maybe even one in the case of the Poisson, but then it doesn't really matter. We've got some, and those parameters are modeled by linear predictors. So the kind of formulas that you have in R or your statistical software. So we have one here for the mean of a Gaussian, and then in the Gaussian, we would also have another linear predictor for the variance or the standard deviation. You can parameterize it both ways. If it was some other distribution, we would have different parameters, but each parameter has its own equation. And you can have the linear predictor or equation where you have certain covariates modeling the mean. You might have the same covariates modeling the standard deviation or the variance, or a different set of covariates that might be important for modeling the variance. And then by way of illustrating how you would practically do this, this is the way it's done in MGCB with the GAMs that I'm going to talk about here. You have one linear predictor, one equation for the mean, and then you have another equation here for the variance or whatever parameter, and then you just choose a family, okay? And this is the location scale version of the Gaussian. Now, not all distributions have an independent mean and variance. A distribution that we're going to have to use in a moment is a thing called the gamma distribution. The gamma distribution is defined for positive continuous data. So it's a bit like a Poisson or something like that, but it's for continuous, not integer values. And as you see here, as we increase the mean, the spread of each of these curves starts to get larger. So this value here has a very low mean, it has a very narrow spread, whereas this blue line here has a high mean and a very large spread. There is what we call a mean variance relationship in this distribution, but we can still model that spread and the mean in a regression model. So let's go back to that data. Essentially, I think it's the same data set, that simulated model that Dakos and co looked at. They have this well-studied, it was a well-studied harvesting, a model of harvesting and overexploitation, where essentially we have some resource biomass X that grows logistically, and then that biomass is harvested according to some equations. So the change in biomass is given by this thing here, and the parameters, there's something in here for the growth rate, the carrying capacity of the situation, the ecosystem, something for the logistic, and then C is the growth rate. And that, if we had only those terms, it would be entirely deterministic, but then we turn it into a stochastic process, so it's got noise through this noise process over here at the end. Right? Now, in the deterministic case, this mathematical model crosses a fold bifurcation when C reaches about 2.6.04. So when it hits that point, the system crashes and we have a total loss of biomass, right? So we know that mathematically that this has it. Now we're using a stochastic version, so it doesn't exactly happen when C gets to that value, but it's going to happen around there, okay? So if we simulate data from this model, this is what we get. And I've cut it off. So I've simulated 1,000 time steps. So we've got 1,000 samples here. And at about, I don't know, 800 or so, the system is collapsing. It's crossed the transition here. And if I showed you all the data, this would be going to 0. And then it would just stay flat after 0. Okay. So imagine these data as being a sediment core where we're starting, this is now old, I've got it back to front, but this is old. And then as we're moving through time this way, so this is the present day, and we're viewing an impending transition because we know it's happened here, right? This is an ideal situation. So we're going to fit 3 GAMs here. We're going to fit a basic GAM that just has constant variance, so there's no model. So we say the variance is the same everywhere. Then we're going to say we're going to have a Gaussian distributional GAM where I have an intercept plus a smooth function of time in both my mean and my variance. So I'm allowing the variance to vary smoothly over time. And then I'm going to do the same thing because biomass can't be negative. So maybe there is a mean, there's some sort of mean, but extra variation beyond the Gaussian that we might want to account for. So we're just going to model this using a gamma, where we have the mean linear predictor, but we also have this shape parameter. We're not modeling the variance directly. We're modeling a parameter called shape. That's the how skewed the distribution is, I believe. And with those 2 parameters, we can actually calculate the variance for the distribution. Right? So essentially, we're saying that each of the observations, a fitted value for each of these data points in here is its own distribution that has some mean and variance. That's what we're doing here. So GAMs, very briefly, the GAMs are a statistical model. They are more complex than linear models, so they can fit data that is more complex than a straight line, but they remain interpretable. They're not like these black box machine learning tools because we can fit the smooth functions and plot them and draw them to see what these models look like. GAMs fit wiggly functions, and these are the wiggly functions that they fit. They're called splines. And splines themselves are made up of lots of little functions. They're not magical. They use what's called a basis expansion. And they are made up of lots of little functions. So that spline I was showing there is actually formed from these 10 little functions. The different colors represent the different functions. This just happens to be a cubic regression basis. And once we've created these little basis functions over our covariate x, could be time, we then choose different weights for the different basis functions. And if we give different weights to different basis functions, we get a different spline. Because we add up the values of all of the basis functions at each time point. That gives us the thick fitted line. Now here, I'm just giving the weights randomly to these splines just to show what's happening. But when we've got data like this, where we've got some underlying true trend that we want to model, but all we know are the gray dots from our data, in that situation, then we don't choose the weights randomly, we choose the weights that make the curve, our fitted spline, go as close to the data as possible. So our regression modeling problem is then finding the value of these weights, our betas, our slopes, if you want to think of them like that, for each of these little basis functions, is we do it by maximizing a penalized log likelihood. The log likelihood measures how close we get to the data. The penalty is measuring how complex, how wiggly our fitted smooth is, and we trade off the 2. And this helps to stop us overfitting. This wiggliness penalty avoids complex models unless they would make us fit the data incredibly well. All right? We have to justify using a complex model. So to recap, what we're going to do, we've got this basic GAM with constant variance. We've got a Gaussian distributional GAM with one of these wiggly functions of time in both the mean and the variance. And then likewise, we've got the same thing here, but for a gamma model, right? Which doesn't allow for negative values. And we're going to see which one fits. So it turns out for these particular data that the Gaussian model had the lowest AIC, so it had the best fit to the data. This is our estimated mean. So this is essentially detrending the data. So that looks entirely reasonable. And then this is our estimate of variance. Okay, and we could compute theoretically what the variance is, I believe, from the math. But throughout, we see this increasing variance. I believe this is just a burn-in problem, that maybe I didn't let the system burn in for enough values to reach equilibrium before I started varying C. Okay, but it should really be flat and then start to increase essentially. Yeah. So our model has done what we wanted it to. We've got this increasing variance out of it without having to resort to ad hoc approaches. So that's good, it works. Let's have a look and apply this to some real data then. This is where Lake 227 comes in. It's an experimentally manipulated system in the experimental lakes area in Northwest Ontario. It's an annually laminated sequence that was called by Peter Levitt, I believe, in 1990. And so we've got annual sediment values from 1943 to 1990. Peter did the fossil pigments, and then he was doing this to compare them with the measured algal communities taken from the lake samples that were sampled every year by the ELA crews. And that's what one of the reasons Peter was doing this work, was to calibrate what the fossil pigment record was showing. But then in a 2000 paper, Kathy Codingham, Jim Ruzak, and Peter, in a paper in Ecology Letters, they showed that for this period before the experimental manipulation and then post the experimental manipulation, that those two periods had different variances. They also showed some other really nice stuff using being able to predict the ecosystem, and showed that the predictability of the ecosystem declined over time because it was becoming more variable. So rather than doing this Levine's test before or after, can we have a continuous estimate of the change in the variance of this ecosystem over time? So these are the data. We're going to use 5 particular pigments. So I'm looking at diatoms and chrysophytes through fucoxanthin, this thing in blue here. We're looking at cryptophytes through alloxanthin, which is this thing at the top. Cyanobacteria and chlorophytes come through from lutein zeaxanthin and also from thiophylline B. And then beta-carotene is a measure of kind of the total algal biomass. And you can see that the system was relatively stable over time. Then they start to put nutrients in, and then all heck breaks loose, okay? And they become much more variable. Can we model that in an ideal system, right? So what we're going to do here, these pigments really do require a gamma distribution. The pigments can't go below 0, and they're all positive, and they have some non-constant mean variance relationship. And because our actual problem included data that were below detection limit, we have to fit this using something like BRMS and a more complex system using the Bayesian software STAN. So what we've got here is our observations. We're saying that they're distributed gamma, so they're going to follow some gamma distribution. They're going to have some mean that's given by one of our linear predictors, and then they also have some shape given by another linear predictor. And each of our linear predictors essentially is going to have one smooth of time, so one trend per pigment. We've got 5 pigments, so there are going to be 5 smoothers for the mean, and 5 smoothers here, 1 per pigment for the shape. So I'm going to model all of these pigments in one go. And I'm going to do that using a special kind of spline from the MGCB package called a factor smooth interaction. Now, actually, for these data, I could fit them in MGCB, the 5 pigments I'm talking about here, because none of those are censored, none of them are below detection limit. And so I could use the Gamels family or the Tweedy LSS family. But in the actual application that had more pigments, we had censoring, and so I used STAN to do that. Okay, so this is what we get when we do that. We have these relatively constant periods early on in the record, and then we have, that's picking up the increase in the pigments after the experimental manipulation. I can explain maybe what's going on in fucoxanthin, I guess, because it seems to anticipate the enrichment a little bit. But it's also increasing towards the end. But more what we're interested in is this thing, which is the variance. And so now it's clear that the after period has more variance than the before manipulation period. But now, rather than just having an estimate before and an estimate after the manipulation, we see we can estimate the variance but throughout time for the whole part of the ecosystem. So the variance was mostly changing in the period during the initial manipulation, and for some of these pigments is essentially leveled off and isn't changing very much. Okay? All right. So we have this continuous measure of the resilience of this ecosystem rather than having a point estimate for 2 periods. Okay. And well, I don't need to have a summary. We can come back to that later if you want, but you've got the slides. Anyway, basically, I think that's really useful to do. And because we have this full conditional distribution, we can look at other indicators, not just the variance. We can look at the skewness, for example, which has also been suggested to be a metric that we might use to look at resilience of ecosystems and approaching a critical transition. And because these models, even if we'd not fitted it with STAN, even if we'd fitted it in NGCV, it's a Bayesian model. We have a posterior distribution. We can do really useful things with that model. One of those useful things that we could do, we get rates of change for the ecosystem. Yeah, I can calculate numerically the derivative, the slope of this line at any point in time. That gives me the rate of change. I can sum that across my community, and I have a community metric for the rate of change, all from a statistical model. I didn't have to do anything else to get that, really. So what am I going to do next? Well, we do actually need to figure out some of the detail. I just said you just sum it up over all species, which you can do. But if you want to compare one lake with another lake, they might have different numbers of species in them. How do you normalize that? So there's a little bit of stuff need to be done to combine variances across taxa to compare between lakes, for example. But then we can, maybe we can also use these kinds of models to generate estimates that are required or that are used in theoretical models of ecology. So there's lots of theoretical work being done on ecological stability and what have you. And it would be nice if we could try and tie some of the values or some of the things that they include in their theoretical models. Can we get them out of our GAMs, our statistical models? Because if we can match up features or things that we can derive from our statistical model, then we may have a nice way of testing specific theories from our paleo data or any other time series for that matter. Okay, but paleo data are not so nicely behaved as Lake 227. Okay, so a couple of things that we really need to take account of. One is, now this happens whether you are using DNA or pigments or anything else. Every slice of mud is going to contain a different number of lake years. And just in the same way that if you take the average of one number, that average is going to be very uncertain relative to taking the average of 10 numbers or 20 numbers, say. If we look at a sample with one lake year, its mean, or that data point, is going to be more variable than a slice of mud that averages 20 years, say, all right? So the greater the number of lake years, the lower the variance. And conversely, the fewer the number of lake years in each slice of mud, the higher the variance. So this is just a property of our data, and all else equal, because of the compression from the water and the sediment, the older samples are going to contain more lake years per centimeter of mud than they do at the top of the core, the mud-water interface. So this implies a trend of increasing variance. And if the accumulation rates have been changing over time, then that just induces a non-linear trend in the variance over time. So we need to account for that, right? And by account, we can do that in a statistical model because we can put that effect in the model. We can include in our linear predictor for the standard deviation or the shape, we can include in there the number of lake years that each observation represents. And I've got a discussion of this in this paper from a paper that I wrote a few years ago, where I include the discussion when we're just looking at modeling the mean, but the same problem applies when you're trying to model the variance as well. In fact, it's even more important that you include this. Otherwise, you might find a trend in variance where there isn't any. It's all due to this sampling artifact. Okay, but there's an extra problem with eDNA, and that's that each sample represents a different amount of effort spent counting things. And so in the sequencer, there'll be more sequences in some samples than others, some library preps than others. And so the number of reads the number of bits of DNA that get read in the sequencer will vary between your different samples. If you spend more effort looking for things, you will find more of those things, and vice versa. So this implies heterogeneity. So it's really important. This is very important if you're doing sort of like diversity work from paleo, and this applies this previous bit. This really needs to, if you're doing diversity, this really needs to be taken account of. Okay, but in eDNA, you've got the lake year effect, but you've also got the number of reads effect. And so I know this is controversial, and there's a lot of literature and people literally fighting with one another about whether you should or shouldn't do this. But I'm just going to be controversial and say you should not rarefy your eDNA information. You should treat them as counts, and then you should do something to account for this extra heterogeneity. And if you want to know why, I'd suggest looking at this paper here by McMurdy and Holmes as one illustration of why. So what we can do in our model, just as we were including the number of late years in our model, we can include something else in our model, which is called an offset, and that's just fancy stats for a coefficient that has a fixed value of 1. We don't estimate it. And so I could include in there, for example, log of the number of reads, and that would be a way of standardizing or normalizing each of my observations for the different effort that we spent counting that piece of mud or its DNA. Right? Now, we can do better than just including the number of reads. There's a package called DESEC2, which will compute for you something called a size factor. And it does this rather than taking the reads, the number of reads for every sample and just assuming that that's the effort. It calculates something that using a robust regression that gives you a size factor for each of your samples. And if you include that as your offset, that's a way to handle this. And then there are other things that you can do, like I was using gammas and Gaussian distributions in my example. If you've got eDNA, you're going to want to use things for counts. So that implies using things like a negative binomial or a Tweedy thing, rather than using Poisson. Typically the variance, the mean variance relationship is different, right? So these are all features that you need to take account of because they're features of your data. They can all be handled in the statistical model, but they make the statistical model a little bit more complicated to apply. Okay, so that was essentially univariate or a few variables. What do we do if we're in the typical situation where we've got lots of taxa? Quantitative paleoecology has this tradition that we typically will view things as proportions. We'll do some dimension reduction. We'll get a picture of our data by an ordination, or we will just plot a stratigraphic diagram. We will do some clustering maybe to group our data by depth. That implies representing the samples through a dissimilarity. And all of these things are approximations to the underlying data, but we do them. And this is what has been taught. This is the tradition of quantitative paleoecology. The problem with that is that if you only ever stick to applying those kinds of models, that, you know, this is statistics from the 1950s and 60s and 70s, then every problem that you as a paleoecologist face is going to look like a nail because you're just applying the same techniques. Now, it doesn't matter, that's not a problem. If you've got the same questions, that's great. But our questions evolve over time. They have to evolve for the kinds of data that we are trying to work with. And they all are questions. The theory that we're trying to work around and work with and develop changes. So that changes the questions, right? So it's okay to follow tradition. Traditional methods haven't just stopped working. There may be better methods now to answer those same questions, but you can still keep using your traditional techniques. But there may be some point where they start to hold us back, where we can't do things, or we can't do things as well as we might do if we had some other tools in our toolbox than just our hammer. So I'm going to talk about something called a topic model now as a way of summarizing complex data before we put it into one of those statistical models that I've just talked about. And I'm going to go through this relatively quickly because I see I'm running a little bit low on time. These are data that Tricia will be very familiar with, but they are a data set from Foy Lake with loads and loads of diatom species here. And the data were actually provided by Jeff Stone because I needed the counts. But I think this diagram is actually using the data that Tricia made available from I think it's the +1 paper that described looking at transitions and regime shifts in this sequence of diatom data. So incredibly complex patterns of change over time. Most of the diatom taxa are actually buried in here in the sort of 1 to 2 to 3% range. They're all down there. And there are some very large things. Those taxa, the dominance of those species has changed over time, a very dynamic system. How can we summarize this data to make it easier to model? I could fit a hierarchical GAN like those things I just fitted to the pigments, but it would be hellishly slow to do it to 100 taxa. And it would be even slower if these were eDNA samples and I had 1,000 ASVs, for example. All right. So we just can't, most of the models that we might like to fit just cannot be fitted to large numbers of samples, large numbers of taxa. So these multivariate autoregressive state space kinds of models, the things that are similar to sort of models that Zofia Taranu was talking about last time, right? They just can't be fitted to this size of data set. Yeah. Well, maybe we're getting to the point where they might be able to, if you want to use a GPU and a fancy computer to do them, but they're not within the remit of most paleoecologists. But we still seek some reduced dimensionality of the data that is going to preserve the signal. Now, we could do that through ordination, but they don't really work so well for these very complex data sets. Correspondence analysis, principal curves, PCA, these things will all get upset to some extent. If you have a long gradient, this might work, but if you have several gradients, this breaks. If you have odd samples, correspondence analysis breaks. And if you want to do PCA on the data, we have to transform the data in some way to get it to even allow it to fit something as complex as that. So now we've got into the process of which transformation should we use, and there's loads of them. Also, the axes of these things, they're a linear combination of all taxa. All taxa contribute to those axes. Some don't contribute very much, but all taxa contribute. It would be better if we could try to partition the data up into groups of taxa that are responding in the same way. So one thing I'm going to think about now is, can we group our species into some associations, some number J, and cluster our samples and our species while we're doing it? And this, hopefully, for those of you who are old enough, will, I don't know, maybe signal to you some ideas from what this software TwinSpan was doing. So this is Foy Lake in Montana. This is Foy Lake, and I guess one of the reasons why it's very sensitive to changes in climate is that there's this very low-lying area here, and this part can dry out, separate the lake into two, or one area might even dry out entirely. And so you can see why the lake might be really sensitive and showing those very large ecological changes. Okay, I am not going to look at this data from any ecological point of view. You can ask Tricia or Jeff about those. I'm just going to look at how might we take that data and simplify it. So a topic model is a model essentially that was designed for organizing text documents. If I gave you all the books in a university library and I went, here you go, organize these books, but I'm not going to tell you really what the books are about. You don't want to have to read them all. I mean, it'd be fun, but it would take you a long time. We could get the computer to do that reading for us, but computers can't read. So what they've developed for organizing text documents are these techniques which try to come up with a mechanism, a simple mechanism by which the computer could envisage the books as having been created, as essentially a random draw of words from particular topics. And then if we can fit our model, it's going to find some topics. So that allows us to organize our documents by topic. And then we can organize the words into topics as well at the same time, so that we don't have to tell the machine, the computer, what the topics are. One of the sort of most widely used things is a thing called latent Dirichlet allocation. But the important point here is it's a generative model. I could sample data from this model and I could get a book. I mean, I wouldn't have one that would make any sense from an English point of view, but it might be a way that the data could have arisen, all right? Now, these things have been used in ecology before. Dennis Villay, who is at University of Florida, had this really nice paper in 2014. And Erica Christensen, Dave Harris, and Morgan Ernest have also a really nice paper from a few years ago in ecology, where they use this and apply it to time series data or to ecological data. So I recommend, if you're interested in these things, go and look at those. Okay. But I'm going to try and explain it now as a community of Skittles, not of taxa, not of ASVs, but of just these little sweet things here. And this is going to represent my bowl of candy, is going to represent one sample. The different colors in here represent different flavors of Skittles, but the candy that I put in my bowl actually came from these different packs. Now I've got 4 different kinds of packs here, but I might have had 10 packs in total in that candy bowl. It was a big bowl of candy. right? So I want to know how many of each of these packs, Skittles, went into that. How many berries versus how many original packs went into the bowl, and then also what is the average distribution of the different flavors in the sours versus the originals versus the berries, and so on. So the individual candies are my individual organisms. The flavor that they are, that is the species, what type of organism it is. And then the pack is sort of these associations, these groups of similarly occurring taxa. So the aim then is to get a distribution for the Skittle flavors within each pack, and then get a distribution of the Skittle packs within each community. That's our sample, each slice of mud, say. Okay, so we get a soft clustering of our samples because our samples don't belong to just hard coniferous vegetation versus tundra versus whatever. They're not in that. They might be, well, this sample is a mix of tundra and pine forest, maybe, things like that. And then the groupings of the species are also soft clusterings because a species might occur in multiple associations, right? Now, you have to tell it how many groups. In here, I've got 4 different packs. If I didn't know that, I'd have to try 2 groups, 2 packs, 3 packs, 4 packs, and so on, and see which one fits the data best. And I can do that using some statistical method like AIC or whatever. Okay. I'm going to skip over a few slides because I see that we're getting close to time, but I just want to go and talk about a little bit the intuition behind LDA. So what LDA, latent Dirichlet allocation, is trying to do is trying to find for each sample, it's going to allocate its individuals to as fewer associations as possible. So each sample shouldn't be made up of all possible bags of Skittles. It should really represent just a few. It's not all biomes, it's just a couple of biomes. And then each association, each group of co-occurring species should really be dominated by a few high probability species. We don't want an association that is basically, well, it's just a little bit of everything, because that's not generalizing anything for us. Okay? So that's what LDA is going to try to do. And we're going to skip over some of the detail here because it's not necessary. Okay. So this is a model that's slightly different. It just allows the associations to be correlated with one another rather than uncorrelated. It's highly likely that this association of species will tend to occur with this association, or this other association will not occur when there's some other association present. We have correlations between our associations. So if we take that into account, this is what we get. So now I've got 10 associations. So I've got 10 groups of species or associations of species in the diatoms from Foy Lake. I've just arranged them over time as a weighted average of depth. So the things that occur later in time are at the bottom. The things that occur earlier in time are at the top. And you can now see in easier detail what's going on. And then we can drill into each of these associations and look at what the taxa are. Okay? And so we've plotted the topic proportions over time. We could ordinate them. This is the kind of thing that we get. We can look at group 1 and see that that's a puncticulata. Okay, we can look at group 2. So that one is dominated by one species. This one here is dominated by some cymbellas and amphoras and things like that. We see that this is very different from these other groups, and 9 is also quite different. So we can ordinate them just as a visual tool now, not worrying about using the ordination and it messing up. We don't have to worry about that happening because we've simplified the data. Now we just use it as a visual tool to look at a much simpler data set. And then once we've got that, essentially we're just using the LDA results as a way of summarizing the data. And then we can take the output from the LDA as new data and model it in the same way that I showed you with the GAMs. And if we do that, fitting a specific kind of regression model called a Dirichlet regression to these data, where Dirichlet, just if we've got percent sand, silt, clay, I only need to know percent sand and silt, right? I can get the percent clay by looking at the difference of 1 minus those. Yeah. Okay, and so this is what we get. If we fit a model to those data, now I've got a summary of how these things have changed through time. I have a statistical measure. I can look at the rates of change of each of these curves through time. I can do everything that I was doing with my GAM before, but I can actually fit it to these data, whereas it would have been very difficult to do it to the full data set. Okay, let's just leave the summary there and the future directions. Yeah, and I will just say thank you very much. And if you have any questions, I'll be happy to take them.
Excellent talk, Gavin, and not just because you used a data set that I'm familiar with. I'm sure there's questions already. I will try to monitor the chat and kind of call on people. Let's go ahead with Luke first, please.
Thanks, Trish. Thanks, Gavin, for a really cool talk. It's really interesting. It's amazing to see some of these methods that are really new to me. My question is, we work very hard in eDNA world to get all these samples, and so that means sometimes we don't end up with a huge number along a time series. We might only have 20, 30, 40, if we're very lucky, across a time series. When do you start getting worried when you're fitting GAMs or that second group of models you were talking about? When should we be worried, and what should we look out for to make sure our models are properly working when we get to low sample numbers?
Yeah, well, I think if you've got that low sample numbers, it's probably overkill to do the latent Dirichlet allocation. I think there are things that you could probably do by just dropping off some of the rare taxa and just ordinating, and then you'd be fine. The GAMs, like the stuff that I had from Lake 227 was just like 50 observations, and that was fitting a relatively complex model to that. So I think that where you start to run into problems is where you try and ask too much. You end up fitting overly complex models. And one of the nice things about the GAMs is that because they've got a penalty in them, we can even make them shrink back to flat lines. So there's no change. We can't estimate anything. And that might just be the answer that you can give. If it's not possible, and I think this happens a lot in paleoecology, where people are using, having to spend a lot of time physically counting the organisms, and it's a very time-consuming thing. You don't always have hundreds of samples per sediment core. We have the same problem. Maybe there are limits to what we can say, given the amount of effort we're willing to put in. And I think being aware of that, maybe doing some simulations on the, you've got some past study that you've fitted